What if it's up on this side? Can you guys hear it better like this? Oh, okay. All right, so it's 8:31 now, so we're going to go ahead and get started. Welcome, everybody. This is the review for Bio 1, BSc 2010C for Professor Yeargain's class. If you're not in Professor Yeargain's class, that's all right. If you're not even here for BSc 2010, that's all right too. Just kind of like be quiet and courteous to everybody else. We're all adults here. We know how reviews work. Also, feel free to ask questions anytime. If you're like raising your hand and I don't see you, just give like a quick like hey or hey Mike or whatever. Yeah, my name's Mike, Michael, one of the two. Either one works. Quick like announcements for Professor Yeargain's class. Don't forget, she also has a review on the 4th from 11 to 12. I'm not exactly sure what that time is, but it is an announcement on web courses, just to kind of like keep that in mind. For some of you, your exam's going to be on Wednesday, and for the others, you get a lucky break and you get to take your exam on Friday. Is there anything else like important that you guys know? So this is like the review that I put together. Some questions may have errors, so please double check it. Don't just like take my word for it, because you just like I make sure that it's like 99% correct. You guys are in charge of that 1%. Is pretty much what I'm saying. Clear? Feeling good? All right, awesome. Now, I believe you guys are going to pass this test, but you guys need to also believe that you're going to pass this test as well. This review has 71 questions on it. I don't think we're going to get through all of them, but the last 15 minutes are going to be like last-minute questions, or like, hey, can you explain this again, or let's go to this question. So like look through the sheet, pick out some of the ones that you really want to go over, and then for that last 15 minutes, we'll kind of hit it up. So, we ready? Yeah. All right, sweet. So, number 1, the number of blank differs in isotopes. What do you guys think? Protons, neutrons, or electrons? Neutrons? Here's some of you saying neutrons. It is neutrons. So, changing the number of protons changes the whole element. And then changing the number of electrons just turns it into ions. Any questions on that? Okay. So like, if there is any questions, just like raise your hand and say yes, so then I don't have to like ask every single time, and then that'll flow easier. So number 2, what is the mass number of an atom? What do you add? D. So protons and neutrons. Great. So what are the 4 essential elements of life? What elements have we seen the most throughout this entire course? Carbon, hydrogen, oxygen, nitrogen. Sweet. So that leaves us with C. So what characters below are those of a hydrogen bond? Now I have a quick question. Is hydrogen bonds an intermolecular force or a bond between atoms? It's an intermolecular force. So although the actual hydrogen bonds, like we think about when we hear like polarity and all that stuff about water and how it's really strong, right? Which one of these is a characteristic of all of them? I saw somebody mouth. It is actually all of them. So think about water. Yeah, like the hydrogen bonds keep it from boiling so like quickly, but also when you pour out water, it just doesn't come out in like a cylinder, right? It doesn't just like plop in your glass. That's not very hydrating. It's not appetizing either. So it is actually all the above. They just like, as the, so like here's our water molecule, right? And then here's, what is the oxygen, positive or negative? Negative? Yeah, it is negative. And then these hydrogens are also positive. So as water's like flowing around it, this is going to be attracted, like positive is attracted to negative. And then like it's just kind of like flowing around, and they're just like, they're breaking and then rebonding, and it's just a like fluid. Make sense? Okay. Which of the following groups contains the largest number of organisms? So this is like the whole classes and domains. So which one of these, like, let's start from the top. Which one's the largest group? Domain, awesome. Kingdom. Phylum. Class. Order. So like another mnemonic, it's kind of, as you can see, like it's kind of hard for me to like just think of it off the top of my head. So what I remember is, do kings play chess on fine green silk? So this is family, genus, species. Now there are a ton of mnemonics online. You could just look up mnemonics for for the groups of organisms. So judging by this, which one contains the largest number of organisms in here? Somebody say kingdom? Did everybody say kingdom? Okay. So yes, it is kingdom. Which of the following is an acid? So just to kind of like rewrite it down. And also feel free to say anything if you can't see what I'm writing as well. So which one's an acid? Is it D? Why would you say D? So awesome. So like 1 to 7. So if you took like, if you had a calculator and you took the negative logs of these, this hydrogen concentration is going to be 14. This one's 7. This one's 9. And this one's 6. And then these are all for pHs. So what's the range for pH of an acid? 1 to 7. Or 1 to 6. 7's neutral. So you have 1 to 6 being an acid. And then which one's the base? 8 to 14. Now let's say that there's, what if they ask like POH, right? What do we do then? Do we freak out? We're like, right? So, like what would be the POH range for an acid? 8 to 14, great. And then what about a base? 1 to 6. So just think about this. So like this would be, this is pH, and then POH is just flip-flopped. Any questions on that? Feeling good? Yeah. Yeah, so pH is one, for like the pH range for an acid is 1 to 6, and then the POH is 1, I mean not the POH, the pH of a base is 8 to 14. Now if it was POH, that's going to be, so 1 to 6 is going to be a base. And then 8 to 14 is an acid. And then 7 equals neutral. Did that answer your question? Cool. So the bonds between hydrogen and nitrogen in NH3 are what? Due to the electronegativity of nitrogen. So is nitrogen highly electronegative, or is it just like a little electronegative? It's highly electronegative. So nitrogen is going to want to pull those electrons towards itself. Now, let's say that, and then what about hydrogen? Is that high, like, do they pull electrons highly to themselves? No, because the number of protons is just 1. So think about like the higher the positive charge, the more negative charge is going to be around it. Positives attract to the negative, kind of like a magnet. So say like I'm going to play tug of war with like a 1-year-old. Who's going to win? Most likely me, right? So like I'm going to be pulling, I'm going to have more of the rope than the 1-year-old. Like the rope meaning the electrons. Does that make sense? Feeling good? Okay, so then what kind of bond is that? Does anybody want to take a guess? Polar covalent, yes. So in between this hydrogen and the nitrogen, and technically it's like this, if we really want to look at it. In between these here is a polar covalent bond. In between these here is polar covalent bond. In between these here. That's just electron pair. Yeah. So it's just there's a polar covalent bond in between the hydrogen and the nitrogen. Which of the following is the most hydrophilic? So what did somebody say? B? Why did you say B? So it does have a lot of hydrogens, yeah. But what does hydrophilic mean? It goes with water. It can mix with water, and it's not going to be like, think about like water and oil. Water and oil don't mix. They're gonna eventually reform. That's because the oil is nonpolar or hydrophobic. So think about hydrophilic, like hydrophobic, like you want to stay away from water. Hydro meaning water, phobic, afraid or away. Philic meaning that it's going to be attracted to water. So what like is water polar or nonpolar? Polar. So what do you think is going to be attracted to water? Something that's polar, right? Magnets. So like we have the hydrogens here, remember? And these are positive, water. But the hydrogens are positive, and then this oxygen is partially negative charge. So which one of these has like a polar bond? See? So H2, if we drew out H2, is that polar? No, because these two hydrogens have the same strength, the same electronegativity to pull the electrons towards it. So actually, it is A. Now here's the logic here. So if you drew out this, so you have CH3, and then it also has an O and an H. And when this is right here, that allows for hydrogen bonding. So this area over here is going to be negative. There's going to be a little bit more electrons over this way, just like how the water is right here. So it's going to be able to mix with water. Does that make sense for everybody? Okay. So what differs among isomers of the same element? What is an isomer? So an isomer is a molecule that has the same, like, they have the same molecular formula, but they differ structurally. So what would the possible answer choices be here then? C. Awesome. So the arrangement of molecules. Cool. All right. So now we get to like a tough one, right? What functional groups are present in the following molecule? You can select more than one. So which one can we kind of like knock off right off the bat that we see? So we do have a phosphate group. And that you could see because you've got this whole thing right here. So phosphate being PO4 2 minus. So you got this whole thing right here just sitting on this carbon. So E is one of the choices. What else? Somebody else want to give a shot? Raise their hand. Yeah. Yes. So there is a carbonyl group. You see here, there's a C double bonded to an O. Carbonyl group. What kind of carbonyl group? Ooh. Is there more than one carbonyl group? No? There actually is. So there's two. And then once I say it, you guys are going to be like, oh. So aldehydes and what's the other one? Ketones, right. So aldehydes and ketones. So ketones are the ones that are in the middle of the molecule. And aldehydes are the ones at what? They're at the end. Awesome. So aldehydes were the one at the end. So let's say this one is just like the central molecule. So which kind of carbonyl would it be? Somebody want to guess? You got a 50-50 shot. It's an aldehyde. I heard somebody say. It is an aldehyde. Just saying like this is the central carbon. So what is this thing then? Awesome. I heard somebody say it's a methyl group. So methyl groups being CH3 somewhere, they could look like this. Like say this is CH3. But instead of like another hydrogen being here, it's a whole nother carbon. It's pretty ugly looking, right? Scary? Ah. Don't be afraid. It's okay. Knowing these and just kind of like having a general idea of what they like actually look like on here, you'll be all right. You'll make it through. Okay. So, what is the empirical formula of carbohydrates? So this is now we're getting into chapter 5 with our like macromolecules and stuff. So who wants to take a guess? A? Why do you say A? What's a common carbohydrate that we've been working with for like cellular respiration and photosynthesis? Glucose, right. What's the formula for glucose? Does anybody know? C6H12O6. Awesome. So then, could we like summarize this? Could we summarize these fractions? How much, like, if we summarize it all, what would be the formula? CH2O. So A is the answer. So you could have like a C100, H200, O100 if you really wanted to. I don't think that's going to actually show up. But as long as you know this, you could say, oh, 100 divided by 100 is 1. 200 divided by 100 is 1. 2. And then 100 divided by 100 is 1. We know that's a carbohydrate. Which of the following does not make up a nucleotide? It's a B. Yes, it is. Sulfhydryl groups do not make up nucleotides. So don't forget about like when we kind of went over transcription and translation and also replication, where the 3 parts of a nucleotide is a phosphate group, a 5-carbon ring, and then a nitrogenous base. So just keep that in mind. So now we're getting into transcription translation a little bit. This type of RNA brings amino acids to rRNA for protein synthesis. Does anybody know? Is it B? Yeah? It is B. So B is known as like transfer RNA off the top of my head, right? Somebody vouch for me? It is transfer RNA. Sometimes like people like to say translation, just transfer. Although it kind of does that more or less if you want to make that argument. But tRNA is what brings the amino acids to rRNA for protein synthesis. Where does protein synthesis take place? Ribosomes. What other like major cell organelle has a whole bunch of ribosomes all over it? Rough ER. Awesome. So what do you think the rough ER does? Makes proteins. So it's kind of like all clicking together in the end, right? More or less. So now, this scary looking thing, what type of lipid is it? It is B. It is an unsaturated fat. So unsaturated has these double bonds right here. Now a saturated one, this would just be a single bond, and then these would have hydrogens on it. Cool? Cool. What type of structure creates the loops and folds of proteins made by R group interactions? Somebody, I don't know. Does somebody want to raise their hand and shout it? Tertiary, yes. Did you say tertiary? Oh, great. Nice job. So yes, it is tertiary. So primary structure is just like what the names of these specific amino acids are. Secondary structure is what? So yeah, it's like the alpha, beta, like depleted sheets and stuff. It's just like how they're folding. And then this is how the R groups are folding, like how they affect the folding. Just don't like get a headache from it. Just look it over, take the stuff as face value to start off with. Then work your way into like integrating. So which of the following nitrogenous bases is only found in DNA strands? Thymine. Awesome. What is thymine replaced by then in RNA? Uracil. Sweet. See, you guys got this. Doing just fine. Which of the following is not a pyrimidine? Oh, I'm sorry. So which is not a pyrimidine? So it is D. It is guanine. So what's the other purine then? It is adenine. Yes. So adenine is also a purine. And then the rest are pyrimidines. So we're going to do a little math. If a DNA sample was composed of 35% adenine, how much guanine is present? 15? So it is 15. If there's 35% adenine, how much thymine should there be? 35%, awesome. So if you did 35 plus 35, that's going to be 70. And then if you did 100 minus 70, you're going to get 30. So that's 30% cytosine and guanine. And then you divide that by 2, and then you're going to get 15. Yeah. Purines. So there's purines and pyrimidines. These three are pyrimidines, and then guanine and adenine are purines. So purines have 2 rings of nitrogen. And then pyrimidines have 1. If you like looked at an actual picture, I think they're on the slides. If you could probably look it up in your textbook as well. You'll kind of see it. The way I think of it is that like the purines are like pure, so they have 2 rings, and then like the pyrimidines are like primitive, so they have 1 ring. I don't know. A lot like that. I just saw that in a video one time. If you look again at like different mnemonics or like memorization strategies, they have a ton of stuff where you could kind of see what works for you. Does that answer your question? Okay. So when comparing plant and animal cells, which of the following is not a unique characteristic to plants? So it's A or cytosol. Why is it A? Do animal cells have a cell wall? We don't. We have cell membranes. That's why we're like circular. So cell walls are actually square, and then you're going to see that in plants, and then we have like a cell membrane, excuse me. Do we have a central vacuole? Yes? We don't, actually. We have normal vacuoles. No, it's okay. So we actually so like they have central vacuoles, so that's why they can absorb so much water, and then they don't explode. Like remember if a red blood cell takes too much water in, it explodes, right? Thinking about like the tonicity stuff. So we actually have cytosol. Is cytosol the fluid or the space that fills up the cell? It's the fluid. So which one's the space? The cytoplasm. So those are very easy to mix up. I mix them up all the time. As I was walking over here, I was kind of like going through my head, and I mixed them up. So just kind of like keep that in mind. True or false? Mitochondria are single-celled membrane organelles that are the site of cellular respiration. False. Why is it false? They have 2 membranes. So if it was true, they are double cell membrane. Can you guys see that okay? Do you want me to zoom in a little more? I'll zoom in a little more. Nice. Okay. So which organelle contains enzymes that can digest organelles which no longer work? It is a lysosome. So lysosomes are filled with like these dissolving toxins that they just kind of like engulf. It could be like vacuoles or just like broken mitochondria, and then just digest them off. Yeah. That's going to be like chapter 6, tour of the cell. Yeah, it's chapter 6. Just take a look at those as well. I think I have a worksheet on my website, but you can also see, I think I do have a video up there as well. So phagocytosis refers more specifically to what type of lysosome breakdown. So there's phagocytosis, and then there's also phenocytosis. So which one do we think this one is? Yes, it is food. So phagocytosis is going to be cell eating, and then phenocytosis is like the cell drinking, and that's when it's like a certain amount. Like it doesn't just like take an entire mouthful of whatever. It kind of like selectively drinks it. Does that make sense? Okay. So sodium-potassium pumps in the cell membrane are great examples of what? Ooh. It is active transports. So think about sodium-potassium pumps. They're pumping them against the concentration gradient. So they want like high concentrations of sodium on the inside, I think, of the cell. It might be the other way around. Double check that. And then they want high concentration of potassium on the outside of the cell. And that's also how your neurons work too, how we're thinking and talking and studying right now, just to make it a little bit cooler and it's not like, ugh. So it is active transport. Which of the following is a characteristic of an integral protein? So what does like integral mean in general? It's like inside, right? So in a membrane, like what would we think like an integral protein is? Inside the membrane. Awesome. So do they bind to the hydrophobic region of the cell membrane? Yes, because think about the cell membrane real quick. We've got the hydrophilic heads and the hydrophobic tails, and we've got two sets of those. So when there's an integral protein, they kind of sit like this inside of it. So yes, they do bind to the hydrophobic region. Can they be transmembrane? Yeah, they can go through. That's how you have like the passive transports and active transports of proteins. They're transmembrane. They're integral proteins. Will they penetrate the nonpolar region of the cell membrane? Which parts of the nonpolar region? Here or here? The outside or the inside? It's the inside. So, do they penetrate the nonpolar region? Yeah, they're inside. They're inside the membrane. So it is D, all of the above. Any questions, comments, concerns? No? Okay. So this one is. For some reason my sheet cut off the question. So, what does it mean for the cell membrane to be amphipathic? So, the amphipathic means thinking about our cell membrane again. You could just kind of ignore the integral protein. That it has a hydrophilic region and a hydrophobic region. So it is actually C. So both A and B. So they have a hydrophilic region, the head, and then the hydrophobic tails. So an animal cell is 90% water and 10% NaCl. Now how I like to do this right off from the bat, I'll just draw a little circle. 90% H2O and 10% sodium chloride. Good old table salt. So now we put it into a solution. We're going to put in a little beaker here. 95% water. So in here is 95% water and only 5% salt. What is the tonicity of the animal cell and what's going to happen after it's submerged? So we're comparing the inside of this cell to the outside. So is it hypertonic, hypotonic, or isotonic? It is hypertonic. So that means that there's more salt on the inside than there is on the outside. So where do you think water's going to flow? Into the cell. Great. So then what's going to happen? It's going to lyse or explode. Just so it is A. True or false? Exergonic reactions require energy to build bonds between molecules. False. So what's the false part of the statement there? So that's what endergonic reactions do. So exergonic reactions release energy by breaking bonds. Does that make sense to you guys? Okay. So false. And then if it was true, it'd be endergonic. So all the following classify a reaction as an anabolic pathway except for what? Heard some people say C. It is actually C. So they do release energy when they don't. Sorry about that. So an anabolic pathway is endergonic. So here, just to accept the accept part, it's not release energy. So what does it do? It takes it in. So it like takes in / build. I'm just going to abbreviate E. Okay. So it's taking in and building energy for an anabolic pathway. Yeah. So an anabolic pathway has a ton of like definitions. It's more of like characteristics of that reaction. So an anabolic pathway is going to have a positive delta G, which means what? What does G mean? Gives free energy. It's endergonic, so then that means also, because we know endergonic means it's taking in or building energy, and it can build large molecules from small molecules. So does that answer your question? Okay. And double check, I think there might be one more characteristic of an anabolic pathway, but, oh no, it's an example. So what's an example of an anabolic pathway that we studied and like had a whole chapter and a headache on, right? Photosynthesis. So think about we're like taking all these small G3Ps and then we're just making a glucose molecule out of it. Don't forget why they do that too. Remember Professor Yergain was like, man, I told this to you guys like so many times and you guys like, you didn't really, like, you forgot about it on the test. And that's okay. I did too when I was when I took this class. But remember that they're like putting in so much energy so then they could get a whole bunch of energy out of it for cellular respiration. They're not like just like generously making glucose for us to eat. Like they don't care about us. They're plants. They want to take care of themselves. So what stops enzymatic activity by binding to an enzyme at a remote area away from the active site? It is D. So it's a non-competitive inhibition. So say like this is our enzyme here. And it's got this weird little thing at the end here. This is where that like remote site is. So when the non-competitive inhibitor fits into this, it's going to turn the protein. It's going to change its shape. And you know how like we know that that these like proteins, like the whole part of proteins is that their shape determines their function. And if we just like threw something in here so it could stop doing its function, it's going to stop it. It's going to change. It's like changing the structure of the protein. Any questions on that? No? Am I answering it okay? What chapter is that, guys? 8 or something like that? So I think it is 8. I can't remember off the top of my head. Most likely it is 8. Because that's like where you get into the whole like cell stuff. So what role does O2 play in aerobic respiration? Does somebody want to say something? I see you guys mouth it, but come on, speak with confidence. Project it. It is E. So it's the final electron acceptor at the end of the electron transport chain. Yeah. The first one, I can't remember off the top of my head. Do any of you guys know what the first one is? No? Yes or no? Maybe so? The first electron acceptor? Well, like the important thing is to know that that O2 forms H2O, right? Well, yeah. So like you break the H2O to get the O2. Okay. But definitely look up like which one's the first electron acceptor. Now, I'm not sure how detailed the final exam is going to be, but keep in mind that there's like 3 questions each on every single chapter, and then like for chapter 17, there's like 8 to 10. So kind of keep that in mind when you're studying. You might not need to know like the very first one, but it's just like one of these kind of like tricky questions that like is important for that chapter. But yeah, just look that up. Also go to a review and ask as well. That's a great question, by the way. So what is the purpose of electron carriers NADH and FADH2? So like remember that electrons hit the NAD plus and then the FAD2 plus, and then where do they go after that? So they do go to the electron transport chain to drop off those electrons, so then we could get all that ATP out of that. Remember the ATP synthase? You got to have the electrons to be able to run ATP synthase. Any questions on that? And also, like if you're having trouble studying for that, the best way is to draw it out. And for some of you that have been to my review sessions and like SI sessions dozens and dozens of times, I'm like, please draw it out, please write it out, watch videos on it. It's gonna make sense. The crash course video, you know how like they do that thought bubble stuff? It's so nice, like with all the colors and it's moving everywhere. Definitely take a look at that. So 32, which one of the following is a substance that is produced from the oxidation of pyruvate? So what is oxidation really quick? Does it gain electrons or lose electrons? It loses electrons. So sometimes like the best way to remember it is oil rig. So oxidation is is loss, and then reduction is gain. So that's just like one way to keep in mind. So this is talking about pyruvate losing electrons. So what is made? What are our options here that we've got? So we've got does it lose electrons and make itself? Does it form glucose? Acetyl-CoA, O2, CO2, which one? It is C, yeah. Awesome. So it is acetyl-CoA. Any questions on that? That's the photosynthesis chapter. Which specific reaction does this occur in? The light reaction. So this is like kind of like the start of it, of photosynthesis. Does anybody have any questions on that? Does anybody have conflicting information on that? No? Okay. So 33. So the light reaction occurs in where? It does occur in C, the thylakoid membrane. So what type of phosphorylation is responsible for the majority of ATP production during cellular respiration? That's a mouthful. So pretty much like what have we seen for ATP production? It's actually one of the steps too that we had to memorize. Did somebody say oxidative phosphorylation? Yeah? Yes. So it is oxidative phosphorylation. which of the following is not a product of light-dependent reaction? So if you guys said D, you're right. So light-dependent reactions are at the beginning of the cell cycle. You don't form the sugar just yet from the light-dependent reaction. You actually form, and this is just like the general oxygen, that useless byproduct, right? ATP and NADPH. That's what you form in the light-dependent. So sometimes, like, the light-dependent is like the dark reaction. I mean, light-dependent meaning the light reaction, sorry. Like, you're depending on the photons hitting the chlorophyll to excite it, and then, is that starting to ring a bell a little bit? Okay. Which of the following is not a product of light-dependent reaction? Oh, we already did that, sugar. So, if a newfound animal, now we're getting into meiosis, mitosis stuff, has a haploid number of 60, what is its diploid number? So first, let's just answer that question. So if it has a haploid number of 60, what would be the diploid number? 120, awesome. So can we cross off any of these here? So we can cross off C, awesome. Can we cross off A? Yeah? Why? Because the N, what does N mean? Haploid. And then 2N would be diploid. So that's just kind of like not even the information that they gave us at the beginning of the question. So now, what would the diploid number of chromosomes be found in? Would they be found in gametes or somatic cells? Somatic cells. So gametes are like what, guys? Sperm and eggs, just like your sex cells. They are your sex cells. So then what does that leave us with? Somatic cells, 2N equals 120, it has to be D. So this one's just like a tricky question, just to throw you guys in for a loop. A karyotype, how many pairs of autosomes does a human have? So karyotype is just meaning like the normal, like the chromosomes that you're going to see in the nucleus. Okay, so that's just kind of like to keep you on your toes a little bit. I don't know how far she's going to go with the jargon of it all, but just to kind of like keep that in mind. Always, 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 always use the textbook and her slides, and also ask her questions too. You can email her questions, believe it or not, like, right? And she also has, I don't know if she has office hours anymore, but she does have that 1 hour review session on the 4th. But like, ask those questions, like, are we going to see what is karyotype? Like, what? But how many pairs of autosomes does a human have? First off, how many chromosomes does how many pairs of chromosomes does a human have in general? 23, great. How many of those are sex cells, like sex chromosomes? 1, so then 23 minus 1 is what? 22, great. Look at that. You guys are on the ball. During which phase of mitosis do sister chromatids separate? Somebody say anaphase. It is anaphase. So don't forget metaphase and like the pre-metaphase too. Don't forget those steps. It's easy to kind of like get mixed up after a while, but you have 2 whole days to review that stuff. You can kind of slowly pace yourself through that. And for some of you, 4 days, lucky. Yeah. Like that? Okay, you're welcome. So now, during which of the following mitotic phases would a cell be haploid? Could I flip this? Yeah? I'm going to flip it. That's not even on there. So which one of these is haploid? All right, that doesn't even work. So which mitotic phase gives you a haploid cell number? None of them. So remember, mitosis, mitotic phase mitosis, you want the exact same copy. You don't want just like some like kind of the same, kind of different. No, you just want the exact same. Like your friend got 100 on their homework and you're like, hey, don't do that. Don't do it. Okay? No, like you just want an exact replica of that cell so then you could do the exact same functions with that cell. Does that make sense? Okay. So none of the above. Which choice below properly matches the stage of mitosis with its description? A. Awesome. So telophase, you do form 2 nuclei. They form right around the daughter chromosomes. So don't forget that jargon too. If she said like, how many daughter chromosomes are in prophase, what's the answer? How many daughter chromosomes are in prophase? None. There's none. So in prophase, you have like the sister chromatids and stuff. But in telophase, or anaphase, sorry, that's when they separate. That's when they turn from sister chromatids to daughter chromosomes. And then they form around that. So also keep track of that as well. So telophase, formation of 2 nuclei. True or false? Gametes are diploid cells such as egg and sperm, and there is only 1 pair of each chromosome. It's false. So like, what's the wrong part of the statement? So they are haploid, actually. Remember how we went to that question earlier? The gametes are the haploid cells. The somatic cells are the diploid cells. Did I say that backwards or did I say that right? Okay. So, which of the following is a source of genetic variation in meiosis? So, it is all of the above. So, what's crossing over? This, awesome. I see some of you like doing it with your hands. So, like, you're kind of like this. Say like these are your, what are these called? Homologous chromosomes. And then they like line up like this. And then they like cross over and then they share genetic information. So then they cross over and then they're like kind of like new chromosomes almost. But like you want that for the genetic variation. What? That's a great question. So what stage does crossing over take place, guys? Prophase? I hear somebody say prophase. Is it? I'm going to double check that one. I can't remember off the top of my head. Whew. So, yes, it is prophase. It is prophase 1. So that's where like they allow the crossing over of non-sister chromatids. Awesome. So independent assortment, yes. Random fertilization, does that, like the chance of being fertilized, doesn't that create some genetic variation? Yeah, it does. Because then, like, it's just like rolling a dice. Like you get a 1 in 6 chance. The mature form of a newly discovered species of eukaryotes contains 12 chromosomes and exists in the diploid state of 2n equals 12. So what I also like during the exam is that you could write on it, but like don't take my word for it. If Professor Yurgain stands up there and she's like, please don't write on this, please listen to her. don't like go up with like a whole like written test, and then she's like, why'd you write all over this? And he's like, Mike told me that I could write on the test. No, take her word for it first, please. So I always like to just kind of like cross off some of this stuff, so we don't need that. We just need to know that there's like a eukaryote that they want us to talk about that has 12 chromosomes, and that's the diploid state. So now in this new species, how many chromosomes are present in a mitotic cell immediately after anaphase? So it's going to be 24, and here's why. So remember, anaphase is when the sister chromatids are pulled apart, and then now what are they called? Said it earlier. It is the daughter chromosomes. Now, is this still, did they split yet? Did cytokinesis occur? No. So is this 2 cells or 1 cell? It's 1 cell. So now you have a cell that has 24 chromosomes, technically. It's not going to be there for long, but it's there. That happens. It's like a kind of like a small little step. Like if you took a picture like froze frame and you said, how many chromosomes are in here? Then you'd say 24. Feeling good? Feeling confident? Ready to knock out this test? More or less. You will, trust me. So, which of the following is the complex of proteins at which the mitotic spindle will attach to the chromosomes? So, what are the, so like when we've got the chromosomes, right? We got this right here. And then the mitotic spindles come in. What are these things that they attach to? Do you remember a little bit? They are the kinetochores. So it's just like this like small thing where they can attach to. I like to think of it like a kinetochore. It's like kinetic means like you're moving. So like a kinetochore is where it's attaching it to move it. Does that make sense for some of you? Also just like kind of like I said, just like keep looking up different ways to memorize that, not just memorize, but like integrate this into your knowledge. Like, so the kinetochores is where the mitotic spindles attach to the chromosomes before they get pulled, like, so then they can pull them apart in anaphase. So which of the following is like true if a normal diploid cell has 8 chromosomes? So what's going on here? Which one is correct? Would it have 8 homologous pairs of chromosomes per diploid cell? No. It's actually no. Are there 7 chromatids per diploid cell? No. Are there 4 chromosomes per cell after meiosis 1? Yeah. So remember, meiosis 1 and meiosis 2, ultimately, you're going to get haploid cells. So remember meiosis 1, you've got like 1, 2, 3, 4. These are 4 chromosomes. And then like when this gets splits apart, then you have 1, 2, 3, 4, 1, 2, 3, 4. That's when it like gets separated properly. Does that make sense? Yes or no? Yeah. Okay. So it is C. So a scientist is interested in studying the event of crossing over. Ooh, we talked about it earlier, right? In mouse gametes. She wants to label some of the mice with fluorescent dyes to better visualize the chromosomes. During what phase of the cell cycle should she look at the fluorescent DNA strands to most effectively study the recombination? Prophase. Which one? 1. Great. We just went over it earlier. Prophase 1 is where the crossing over. You could do like, here, I'll do this. It's like where the crossing over occurs. Yeah. So it is B. Which of the following, Which of the following is true for both meiosis 1 and meiosis 2? I kind of gave this away a little bit ago. It is A. So both of them produce haploid cells. So just to kind of like go over like what it's not, why is it not? Do sister chromatids separate in anaphase of meiosis 1? I mean, obviously it's no, because I told you guys the right answer, but what separates then? Great, so the homologous pairs are the ones that separate. So we could just like cross this out if you guys have it writing, and then just write homologous pairs. So instead of studying like what's wrong, we could just literally like cross out this study guide right now and start writing like we have our own notes set up. And also use that checklist as well that she posted on web courses. I've seen people just like put some gaps in between each word and then they just write like general notes on them, and that helps out a lot. Where two alternatives for a trait are tall and short and tall is dominant, the phenotype of a heterozygous individual would be expressed as what? So what does it mean with like it's a heterozygous individual? Great, so you have like a big letter, so I'll just use T, and then a little letter, which is T. So tall, looking at this information as well. So we have like one trait, you have a tall trait and a short trait. Tall is dominant. Which one's going to be expressed? The tall, great. Oh, I guess I could have, should have, would have used S's instead of T's. But don't forget, sometimes it's hard to see which one's S when you get to deal with the sex-linked traits. So just stick to a letter. You don't need to like come up with a new one every single time. You can if you want to. I've always used like A because just easy, first letter, all that stuff. So when Mendel crossed his F2 generation of purple and white pea plant flowers, the dominant to recessive ratio was what? It's B, so 3-to-1. So remember the F1 generation, what were those called? Those were called the true breeding plants. So that was when true breeding means like that they're homozygous, either dominant or recessive for both. So if you put those in a Punnett square, right, and you got this right here, and then you got these, this is a little t and that's a little t, you're going to get heterozygous all across the board. So these, right, the true breeding plants were his F1 generation. And then his F2 generation was right here. Now, when you cross those, and I know like some of you may have just kind of like glanced this over, and then others like actually wrote it out. But when you write out two heterozygous individuals, that they're going to have the same ratio no matter what the trait is. It's going to be the same. Now, I'm also speaking of homozygous, I mean not homozygous, for like monohybrid crosses right now. But like here, you could see that 3 display the dominant, that's the 3, And then 1 displays a recessive, 3 to 1. So that's just like really quick and easy. If you saw something that said like, obviously read the whole question, but if they gave both of them for 2 of the when you cross 2 heterozygous, that they're going to have that same ratio of 3 showing the dominant trait and then 1 showing the recessive trait. Now that's the phenotypic ratio, but what would be the genotypic ratio then, guys? 1 to 2 to 1. So 1 being the homozygous dominant, 2 being the heterozygous, 1 being the homozygous recessive. So in the cross of NN, with this cross, what proportion of the resulting F1 would be homozygous dominant for both genes? So now we're just talking about like what that first cross is going to be. So here's where we get into, remember the 2 to the nth rule or FOIL. I prefer FOIL. You just kind of like get to see it instead of like dealing with exponents, which can be scary sometimes, like it is for me, unless I have a calculator. So using the FOIL, you're going to do first, outer, inner, last, and like the first, outer, and inner, last. Now when you do those together, you're going to get MN, 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 MN for this parent right here. And I'll use a different color for this one. And then you have MN, 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 MN. Now, we want to save some work, right? We're trying to get this test done. We want to get out of there. It's 8 o'clock in the morning. Like, why am I taking a test this early, right? No, that's fine. We don't need to, like, to keep putting these into the Punnett square for a dihybrid Punnett square. We don't need to do that. We could just see this and be like, oh, hey, this is what the offspring's going to be turning out to. So if we do MN, that's an N, and then an M and an N, we cross these together, you're going to get MM, NN. Heterozygous for both. Does that make sense to you guys? Do you want me to do that again? No? Yes? Okay. Here, I'll do it on this paper. So let's say, so they had the genotype MM. So they were homozygous recessive and homozygous dominant for the other. And then this one was just the opposite. So when you FOIL it, you do the first, outer, inner, last, and you're going to get, so you're going to get little m, big N, little m, big N, little m, big N, little m, big N. Good so far? Okay. So then you do the same for this one over here, right? And you're going to get the same stuff. Big M, little n, big M, little n, big M, little n, big M, little n. But instead of like putting this into our 16 Punnett square, I know it's like not good. We could just look at this one and be able to determine all of these other ones because they're the exact same gametes that are formed from all these right here. If we really wanted to, we could do it, but we're going to end up getting heterozygous for both. Does that make sense? Cool. So Marfan syndrome is a dominant single gene defect. It's characterized by lens dislocation, long limbs, spindly fingers, caved-in chest, and a weak aorta. This is an example of a blank trait. So now we're getting into like the variations of Mendelian genetics. Which of the following would that be? And now a couple of you guys are like, what? Like, I completely forgot about this. Don't worry, it's okay. It's good to be wrong here and to like catch yourself here than to go in on the final exam day and be like, what? I forgot about this. Like, I'm here, I'm like throwing these curveballs at you guys so then you guys could catch up. So, with that being said, which one of these are they? Is it B? It is B. So pleiotropic means that, I'll put this over here. Here's what I've got. So pleiotropy is a single gene influences multiple phenotypes. So, and then you also have polygenic inheritance, which is kind of like the antithesis of it, is multiple genes influence a single phenotype. So like that's how I like tried to associate those the most. Remember how like we have to memorize like cytoplasm and cytosol, like which one's which. That's how I remembered those two, is that pleiotropy is like the single gene affecting multiple phenotypes, and then polygenic inheritance, I like to think of it as, since it's two words, you need multiple genes to affect one phenotype. And that's when you can kind of get into epistasis with polygenic inheritance. So a homozygous red and a homozygous white flower are crossed, and the offspring are all pink. What is this an example of? It is A, incomplete dominance. So what is codominance? Yeah, awesome. So that's when they like both show. That's how, remember how she's talking about like the tabby cats and stuff where they're like black and orange, or sometimes they're called calico or that stuff. Pardon me. So incomplete dominance is when it's just a blend. So that's like, so I just like to think like if you mix red and white, they're going to come out pink. Now if it showed like, now if they said something like if you looked at it a little bit more closer, then it showed like the white and the red right next to each other, then that'd be something different. That would be codominance. But they are coming out all pink, so then that means that it is incomplete dominance. Any questions on that? No? Okay. So, oh, you guys can't see that. Here we go. Which of the following explains why XXX females usually go undiagnosed? So this is when they have 3 X chromosomes instead of, what's the usual one? 2. Great. So what would we think it would be? It is B. So remember that for females, one's activated, one X chromosome is activated while the other X chromosome is inactivated. Now what about males? Do they have one that's like inactivated? No? What do males have? They have an X and a Y. So if you disable the X chromosome, then we just have a Y, and then most likely they're going to die. So, or at least like the cell would be. We don't want that. So all but one can be inactivated in the XXX, so like you just see that phenotypically and like characteristically they're good. They're able to function. Now what is the definition called when, like what is the inactivated chromosome called? Inactivated X chromosome. It's a bar body, right? So a bar body, do I have it in here? I have it somewhere. Okay, well, look up the definition, but a bar body is the inactivated X chromosome that isn't expressed. So now we get into more genetic stuff. So black hair for horses is dominant over brown. However, when a homozygous dominant haired female is crossed with a homozygous dominant male, a few offspring show a whitish/tan color. What could be the explanation? A. Great. So it is epistasis. So the definition of epistasis is, and I have this one, it is two or more genes interacting and masking the phenotype of another gene. So remember during like the many problems that she had, she'll have something like golden retrievers or like the horses. Oh, could you guys even see that one? Yeah, okay. So it is epistasis. So epistasis, I like to think that's the one that starts with an E. So it erases, doesn't actually erase it, but it erases like what the other one would be expressed. Make sense? Okay. So a homozygous red and a homozygous white flower are crossed, and the offspring are all pink. That's the question that I already had. I'm sorry about that. But what is it? It's A. So it's incomplete gut dominance. Now let's say, you know what? I'm going to change it. So they're going to be are spotted. So make that correction. So what is it going to be? Codominance. It's going to be C. Yeah. Okay. Well, I'm sorry about that, guys. What is the purpose of topsomerase during DNA replication? And that's like, whoa, now it's really like, whew. Right? What does it do? It is it C? What do I have? Yes, it is C. So it cuts the phosphate backbone ahead of the replication fork, so then it prevents the DNA from getting overwound. The whole thing with DNA replication, it has so much proteins and enzymes that it doesn't want to just be like tangled in a jumbled mess. Like if you tried to untangle some string and then you're also trying to like recopy that and then you're also trying to like you're going to need some help. You're not going to be able to do that all by yourself. You're going to grab some friends and be like, hey, I got to copy this strand of DNA. And then they're like, all right, I got you. I'm going to cut down ahead of the replication fork to prevent the DNA from getting overwound. And we'll call that friend topsomerase. Now, if that just like flew over your head, I'm more than happy to, one, re-explain it. And two, don't forget, it's also pre-recorded. So you guys can come back and look at me re-explain stuff again. Technology, right? So 57. Which of the following refers to the correct model of DNA replication, where a newly synthesized strand of DNA is half the part of the parent strand and half the newly synthesized? It is A. So it is semi-conservative. So think about it this way. In the end of DNA replication, you're going to have one that was the parent/template strand. Now we're getting some more vocab. And then we're going to have another that's the new strand. That's going to be the new strand made. But then don't forget that for like you're splitting up one and you're getting two. So there's one over here that's like kind of the same. It's the same thing. So it's semi-conservative because you're saving half of the parent strand, but you're just making like new stuff out of it. You're not just like copying this and then you're just throwing it away to recopy this new strand. Like you've got perfectly good DNA here. You might as well make some use from it. Does that make sense? Okay. I'm just throwing like ways to kind of keep it sticking in your head. Now the best way, and how like I've come up with all these different ways to like keep a stick in your head is to study in a group. Look around you. All of you guys are trying to pass this exam with flying colors. All of you guys want to like get a good grade. Trade numbers. Even if you might, like some of my best friends that I have, I met up with like studying. So that's like it's gonna work out in the long run. You'll be able to talk, throw out new ideas, be able to be like, I don't really get this. Like being able to answer these questions really quickly, it helps out in the long run. So after I'll stop my speech there. So true or false? DNA polymerase can only add nucleotides to the 5' end of the DNA strand. It's false. What does it add it to? The 3'. Great. So if that's the only thing that you remember from the replication, that's just fine. That's like it helps out so much if you have to speculate on something. Yes, you add it to the 3' end. So now I have a question. Everybody's like trembling, right? Oh no. So now we've got this template strand here that's running 5 to 3'. And let's say that the origin of replication is right here. So like where is the origin of replication? What is like the name? Like if you broke down the name, what would it mean? Yeah, it's the beginning of what? Of replication, right? It's simple. Like don't try and overthink it. Take stuff face value, then integrate it. That's the origin of replication. So where is DNA polymerase 3 moving? It's going to be moving this way. So can it add it to the 5' end or the 3' end? The 3' end, right? Well, so then now we get to this problem, right? It can't really move this way. It can't do that. Why? Because it can't add it to the 5' end. If you were trying to do the new strand, remember it's anti-parallel. Remember that anti-parallel rule? Just meaning that when there's a 3' here, there's a 5' here. And when there's a 5' here, there's 3' there. Simple. It's not going to want to move this way. It's adding, and it's like a mistake that I've made a million times over. It's not adding to this new one. It's to the template. It's not adding to the template strand. It's adding to the new strand. So it needs to go in this direction. So I'll kind of like draw that a little bit better. So like here's the origin of replication. And then we had 5' here, 3' here. And then you've got this one 3 to 5. It's going to need to move this way, right? Can it just like go all the way down here, or does it move in steps? It moves in steps, right? So what is the name of this strand then? Awesome, I heard somebody say it's the lagging strand. Now let's say if we looked on this other side of the replication, origin of replication, there's a 3' here and a 5' here. 3' here, 5' here. Where's the DNA polymerase 3 moving? To the left or the right? It's to the left. Oh, it's kind of backwards for you guys. No, it's right. So to the left. It's moving this way. So it could keep up with the helicase, like cutting apart the hydrogen bonds. So then, is that one the leading strand or the lagging strand? It's the leading strand. Does that make sense to you guys? Okay. So true or false? The two sides of the double helix structure of DNA are held together with weak hydrogen bonds. True, I just said it. Yes, so the nitrogenous bases are connected together via, or by, hydrogen bonds. Pardon me again. So DNA, okay. Does that make sense for everybody right now? Cool. DNA is the genetic material of all living organisms. Which of the following describes the difference between prokaryotic DNA and eukaryotic DNA? So, is this a back? Yeah, it's on the back. So, just remember this one. Is it this one? Is eukaryotic DNA anti-parallel while prokaryotic is not? I'll give you a hint. It's no. So, like DNA in general, the anti-parallel rule, one moves 5 prime, one moves 3 prime. So we've got these 3 left. So which one could it be then? It is D. So prokaryotic DNA is circular, while ours is linear. And then we've got like the double helix, so it really is like this. Right? Don't look at that. But what's nice about the prokaryotes, and that's why like we have to memorize this like really ridiculous DNA replication. Like I wish we could just memorize the prokaryotes, right? Yeah, no. Okay. Well, anyways, they could just move in one whole circle and then they'll be done. For us, we have to like break it up into segments and like multiple origin and replication. There's like all this stuff. It's not a slow process. Like it doesn't take you 10 years to heal a scrape on your hand. It's pretty fast and efficient too. So DNA polymerase and RNA polymerase have similar functions. Which statement is an incorrect description of these enzymes? So it is A. So both of them do not need a primer. Does that make sense? More or less. Yeah. It is DNA polymerase 3. So remember in the lagging strand that DNA, they have to put, where's that drawing again? So remember in the lagging strand, it knows where to start because they put down a primer. They'll just put like a little bit of a small segment of RNA nucleotides, and they'll put it like this. So then DNA polymerase can like say, oh hey, I got to start here and keep going back, and then they'll come off. And then they'll be like, okay, I got to start back here again. They'll keep coming back, going off. Keep going back, going off. Obviously, it's not just like one doing an entire strand of the replication, but does that answer your question? Okay. So it is A. Both enzymes do not require a primer. But what does RNA polymerase need though? What do they need, like to start? It's the Tata box or the Tata box, or it's just like it's like the transcription initiation complex is another way that it's said, but the Tata box is like kind of like where it starts. Yeah. So the codon chart shows that there's a redundancy but not ambiguity in the genetic code. So in other words, what does redundancy mean? It's like more or like repeating. So if you remember like looking at that chart, now remember you don't need to memorize that chart, luckily, right? But you do need to like be able to read that chart. So definitely keep that up. So there's not ambiguity. So they don't have like one codon. Let's say it's like AUG. Which one's that one? Start. Awesome. What is it also called? Met. So there's not like AUG and then it doesn't code for like 4 different proteins. No, we know that the AUG is met or start. So which one, how could we like explain that? into like words or an answer choice? Heard a couple of you say it. It is C. So individual amino acids can have more than one codon, but each codon only translates to one amino acid. Feeling good? Okay. Which enzyme is essential for transcription and has functions such as unwinding the double helix and synthesizing mRNA? So, what can unwind the double helix and synthesize mRNA? Anybody want to take a guess? You got a 1 in 4 chance. 1 in 3. Yeah? No? What if I called on one of you? Right? No, I'm just kidding. So, it is RNA polymerase. Yeah, so RNA polymerase, unlike DNA polymerase, can unwind the double helix and synthesize mRNA at the same time. Because you're just kind of like, think of it like you're like writing like a letter or something, or like you're writing a speech. Like you could write out the whole speech and then like you take little notes on it as to remind yourself what you want to say. So it is B. In eukaryotes, we can make pre-mRNA that must be edited before releasing into the cytoplasm. So what do we do to prepare this like pre-mRNA? There's like a lot of stuff, right? I kind of went over it on Friday if you were in the 9:30. Michael might have gone over it as well. I wasn't there for that though. Anybody know? Is anybody going to take a guess? Is it A? So is A one of the steps? Does it add a this is also called a poly-A tail. So if that kind of like so I picked some words that might like that stand for something that you guys have already seen before, but like just showing you what it could be. Not saying that it will be that, not saying that it won't be that, but just kind of like reminding or like showing another way that it could be seen. So A is one of the answers, but could C also be one of the answers too? Could B also be one of the answers too? Awesome. So it is D actually. All the above. So I just have a question. Well, it kind of already says it in here. So which one is introns or exons removed? Introns are removed. And then exons are the ones that are kept. I like to think exons are the ones that exit the nucleus to go and be replicated. So exons are just exiting with the mRNA. At which site of the large ribosomal subunit does the first tRNA bind to? It is P. So if we looked at the ribosome, and we've got like these like 3 little like window looking things, there's A, there's P, and then there's E. These are 3 different sites on in the ribosome. Might be on the ribosome. Not exactly sure. Actually, it is inside the ribosome because they kind of like the tRNA goes in like putting a dollar bill into the vending machine. So first, like the vending machine, you don't just like put it right here and it just starts sucking it in right away. I'll put it in like you gotta like it's gonna take a little bit before it starts. So what binds to the P site? What's the thing that we remember that came up earlier here? The 3 nucleotides. It's the OG, the MET, the start. That's going to start on the P site. And then then it's going to like the anticodon is going to fit in here, and then it's going to code for that amino acid, and then they start binding to here, and then when they exit, then they fall off, and then you just get left with a polypeptide chain. Making sense? A little sense? If it's nonsense, let me know. Ooh, so a tough one. We got a pedigree. So here's what we can look at right now. The following pedigree shows a recessively inherited disease with filled-in shapes indicating individuals who have the disease. What are the genotypes of the different family members? So now that's like a whole lot of information, but remember that we could possibly write on this test, right? So a recessively inherited disease. Does that say anything about being sex-linked? No? So what is it then? Is it sex-linked? No. So if it doesn't say anything about being sex-linked, it just says that it's a recessively inherited disease. They could say that it's a recessive autosomal disease. Then it's not sex-linked. We don't need to deal with like the XY stuff. Just in case, what are the squares? Who are the squares? Males. Males are squares. So what are the genotypes of the different family members? So we could start, like this is for the whole tree. So let's say I want to know the genotype for number 5. So this would be, like this is just like all the different numbers, but it could also be like generation 1, generation 2, and generation 3. That's a 3. And then also it could be like these would be like 1, 2, 3, 4, and then 1, 2. So I'll let you guys do this whole pedigree like on your own, or like when we get into like a little bit of the to the question and answer period. But what about number 3? What is the genotype of number 3? I saw somebody mouth heterozygous, but why? Great. So like one has to be dominant because it's not filled in. But the way to tell that if they're heterozygous or not is you do a test cross, right? Remember the test cross with the pea plants? Is that when 2 heterozygous individuals breed, then you can, there's that possibility that you're going to get a homozygous recessive trait. So like when you look at, you could look at this and say, okay, these 2 parents had a recessive child, so then that means they must be heterozygous because they don't show the disease, but after like their gametes fuse, they like have a child that has the disease. And then that's the same logic here. These 2 bred, and then they had a child. Now what about this one? What if they asked this? What would that genotype be? Do we have enough information about it? No, we don't, because we are able to see that, but so we would just write it A blank. So we don't know that stuff yet. Any questions on that? Did you guys follow my logic okay? Yeah? Okay. So how are you guys feeling about the like test crosses and stuff? You guys feel really good about that? Because these last like 6 or 5 or so are all test crosses and like pedigrees and that stuff. So we did go through the whole study guide. Now we can, if you want, go through these. The answers are posted on my website though, which will give you the option to kind of like see what happened. And also like, yeah. Yeah, sure, which one? Number 70? All right. So in eggplants, there are true breeding deep purple eggplants. And for some reason, the superscripts didn't work out too well. So it really is supposed to look like CP, CP. It's supposed to look like that. And then these ones are CW, CW. So if you breed a purple and a white eggplant, you get a lavender pale purple eggplant, which is the CP, CW. If you breed these lavender eggplants with each other, what is the expected colors and ratio of their offspring? So now I'm pretty much asking, with the F2 generation, hint, hint, wink, wink, what's going to be the ratio for the genotype? Does anybody want to take a guess? You want to shout it? Yeah? It is 1 to 2 to 1. So genotype, this is 70. Geno is going to equal 1 to 2 to 1. And then pheno is going to equal 3 to 1. Do you see that? So these right here, they're heterozygous, because here the white eggplants and then the purple eggplants, when you crossbreed them, they turn lavender. So this is talking about what kind of trait? Like what's being shown here? Yeah, incomplete dominance. So incomplete dominance is being shown, so that's why like the white and the purple, when they mix, you get like a light purple or a lavender. So if you did CPCW, that's kind of rough here. I'll do it over here. So CPCW, CPCW. And also, if some of you guys are like leaving early, don't forget to like leave your review sheets too. Thank you. So CPCP, and then CPCW, CPCW, CWCW. Does that make sense? And then you have 1 to 2 to 1 for genotype, and then you're going to see, oh, I caught myself there. So the phenotype, since it is an incomplete dominant trait, you're still going to have 1 to 2 to 1. Thank you for making me do all that. So for some of you, like, did you follow that? Because the phenotype's actually different because it's an incomplete dominant trait. So I'll definitely need to, did I put that wrong on the, no I didn't. I just wasn't looking at my sheet. So, yeah. Does that make sense? All right. Do any of you, so like right now we just hit 10:14, so we have 16 minutes left where I can answer any questions you have. We could do a couple more of these. We can look at another question again, and we could kind of like come together, or we could just say like, what is, like, do we want to run through cell respiration again? Do we want to run through photosynthesis again? Do we want to run through transcription translation right now? Like, what's going through your guys' head? What do you guys have questions-wise? Do you guys have any specific questions? Yeah. Yeah. Oh, did I say A and C? Oh, I'm so sorry about that. So A and G, I think the reason why I was saying that is because the answer choice was C or something. I don't know. But okay. So to like backtrack on that, thank you. A and G are the purines. And then C and T and U are the pyrimidines. And that's for nitrogenous bases. And that's like what you see in your DNA that like codes everything and anything about us. Yeah? Cool. Thank you. Anybody else? Anybody want to? Okay. Okay. So yeah, we could go over the stages real quick. Here, we'll use this sheet right here. So I'm not going to go into super great detail, but I will orally kind of like talk about it. So first we have mitosis. That one I think is a little bit easier to start off with if you guys are having a little bit of trouble with that information. So what about mitosis do you guys, like what's the start of mitosis? Like what's the first phase? Prophase, awesome. So like another great way to remember it is PMAT. Remember that? So prophase, metaphase, anaphase, telophase, telophase, either one of those. So like these are like the four main steps, and then you also have cytokinesis, that's an E. So prophase, metaphase, anaphase, telophase, cytokinesis, that is where you, like, those are the four main steps. Are you guys familiar with those? You guys feel good? So like prophase, I'll just kind of get it on the record at least. So prophase, you have, where is it? All right, awesome. So prophase, you have the centrosomes start making the mitotic spindles, and then you also have chromatids condensing. Now don't forget, like, you could scramble to write this down, but you also have it on the YouTube site as well. So, and then there's like prometaphase as well, that's blended into prophase. You have like the nuclear envelope disintegrating. The centrosomes are now at opposite ends of the cell. Kinetochores, the microtubules attached to the kinetochores, and then the microtubules start like elongating to lengthen the cell. And then metaphase, you have the metaphase plate, the chromosomes line up there. Anaphase, that's when they separate. And then telophase is when the nuclear envelope forms around them. And then cytokinesis is where it splits. Okay? So like those are like the four main stuff. It's good to kind of like, now if you look at the slides or if you look at the textbook, you're going to get like a whole bunch of information, right? You're going to be really confused, or like you're going to like look at this and be like, I'm going to have to memorize all of this. Pick out some of the main parts. Pick out the stuff that sticks out to you the most. Be like, okay, I can remember prophase as the nuclear envelope disintegrates and the chromatids condense. And then you can look at metaphase and say sister chromatids align at metaphase plate. And then you could look at anaphase and then you could say the sister chromatids are pulled apart and now they're daughter chromosomes. And then telophase, the nuclear envelope forms. Cytokinesis, the cell splits. That kind of stuff. That's just kind of like how to study and memorize that. Yeah. Say that again? Awesome. So, yeah. So cytokinesis. Yeah, that's like when that's when the cell nucleus is formed. Telophase is when it starts to form, but it's not really like complete yet. Also, like if you look at a whole bunch of different videos and stuff, I know I like to preach that, but it really does help in the long run. Other than like trying to like read the slides alone. Would you guys like to hit? You guys want to hit meiosis as well? Do you guys want me to hit that as well? Feeling good with that, more or less? Yes? That's what I'm going to do. So, for meiosis, it's nice because you have the same PMAT but it's 1.
So this is meiosis 1, and then you will have meiosis 2, which I'll do in a different color. So P1, M1, A1, and, 2. These are 2's. Oops. So P2, M2, A2, and T2. And then, like, the cytokinesis as well. So that one, you got to have to, like, studying-wise, look at it the same way. Just look at, like, some of the major things where, like, you could separate prophase 2 from prophase 1, like, looking at those differences. I have a question for you guys. Does the interphase occur between telophase 1 and prophase 2? No, it does not. Because then if you, like, think about interphase, remember you have G1 where the cell grows, G2 or S phase where that's the replication. That's chapter 16, what we went over. That, like, that whole process, you know about S phase. Remember, it's all kind of like linking together in the end. And then you have G2 phase where the cell's growing again, prepping for cell division, for mitosis / meiosis. And then it goes through mitosis and meiosis. So you don't want to reduplicate these chromosomes, because then that just, like, bring it back to diploid and then it'd go back to haploid again. It just wouldn't really make sense. So, like, just to kind of, like, show you, at the end of cytokinesis for meiosis 1, you're going to have two daughter cells, and then, like, let's say we'll give them 4 chromosomes. So what would be, if this, is this the diploid or the haploid number? It's the haploid number. Great. So what would be the diploid number then? Just looking at one cell. So is the diploid number 4 or the haploid number 4? The haploid number is 4. So then the diploid number would be 2 times 4, which is 8. So you're in the right mindset. But, like, yeah, and then, so then we go through meiosis 2, and now you're going to get, 4, and remember that these non-sister chromatids are separating. So, like, and let's say, like, the beginning cell looks like this. So ultimately, know the steps in between those, but that's, like, the end product stuff. Now, she did say that she's not going into, like, super nitty-gritty details. She's not going to, like, go, like, she's not going to go, like, super hardcore on you guys. Like, she wants, the whole point of a final is for her to test and see that you got out the information that she wanted you to get out, for you guys to, like, grow when you go into, like, Bio 2, or, like, when you go into Biomed, any of that stuff. Like, this stuff, it's not going to come back to haunt you, because you guys have this information. You have the time to study. You just need to, like, you're going to put in the work, and I know that you guys are going to do just fine. Like, you just got to, and then when it comes test day, you got to believe that the work that you put in is going to put you on top. You also have to think about that too. You have to believe in yourself and not, like, doubt yourself all the time, because then you're going to spend, like, 20 minutes on one question, and then you're going to run out of time. So the best way, kind of like taking this test-wise, you might not think it's a lot of time, but we've been taking tests that are like 30 questions in 50 minutes. Now we're taking a 50-question test in an hour and a half. So we're just getting, like, 20 extra questions, but we have, like, 3 times the amount of, or, like, double the amount of time to take that exam, almost. So
just kind of like to put that mindset in your head. Don't sweat that stuff. You've got it. You've also got the 2 extra bonus questions where like you can get 2 wrong and still get 100. Like some of you might be like, no, that's not gonna be me, and then you're gonna like pull up your grade and you'd be like, wow, that's me. Like it's gonna happen. You guys are gonna do just fine. Besides my little pep talk I just gave there, I didn't mean to. That was like some just motor action stuff there. But like, what other like content questions could I answer for you guys? Yeah. What's the haploid number at the end? That's a great question. So don't necessarily just look at all 4 cells. When they talk about haploid number, they're just looking at this. Like when they give the diploid number, do they give the amount of chromosomes in every single one of our cells for our entire body? No, that'd be like billions. I can't remember the number off the top of my head. If you want to look it up, do it. But no, so they're just looking at 1. So how many chromosomes are in here? 4. Great. How many chromosomes are in here? 8? Ooh. I was a little bit indicative there, so you might doubt yourself now. But it is actually 4. So like to kind of like look at this, the sister chromatids are this little part, while this whole thing is the chromosome. Does that make sense to you guys? So like, if you separated them, that's a sister chromatid, and that's sister chromatid. Obviously, if you separated them, they become daughter chromosomes. Yeah, I think I know what you're saying. So like, because this thing and this thing are going to code for the same expression, the same gene expression. Is that what you're trying to say? Yeah. So like, that's why when you want the exact same cell, and then like the daughter chromosomes or the sister chromatids, the sister chromosomes, daughter chromosomes. Anyways, look that. I don't know, I'm just like jumbled up in my own words, but yes, that these 2 X's, the 2 chromosomes are going to code for the same thing. We've got 2 minutes, so any like last minute questions or concerns, like is there like a certain definition that you guys might have tripped yourself up? Is there something like itching at the back of your head? Yeah. Yeah, what number is that real quick? Number 10. Oh, 8. Yes, I see it. This one right here? So hydrophilic meaning that hydro meaning water, philic meaning that it likes water. It wants to be with water. What determines whether it's hydrophilic or hydrophobic? Just remember this. Polar molecules want to be like a magnet. If you put 2 magnets together, are they just going to sit there? No, they're going to connect with each other. That's what polar molecules are almost. Like they have a partially positive and a partially negative end. So they're going to orient and kind of be attracted to those partially positive, partially negative ends. That's why water has such a high boiling point. That's why water has like all these properties where we and all of the plants and every biological organism can survive from these awesome the hydrogen intermolecular forces because of the polarity. Does that make a little bit more sense? So then when you get to this here, you have to kind of like pick it out and then like looking at the electronegativity. Now I do remember that she does have some electronegativity stuff on her slides, but also kind of like look in the textbook as well. Because if you've noticed, if you look at the slides, like sometimes the definitions are like they're not there, but the word is there, and you're like, what is that? But like if you have the physical textbook, you can have like the slides and the textbook with you. If you have the online one, you can just put them in 2 tabs and like look at it that way. But it is I'm out of time, but really quickly, it is CH3OH because there's this OH molecule here bonded to this C, so then that makes it like polar on this end.
So that's why. Now, if you look at these other ones and draw them out, they're not polar. And the polarity is determined by the strength of electronegativity of one atom between another one. So thank you guys so much for coming. If you guys have like the sheets, could you please